Assalamu alaikum and a very warm welcome to everyone. Please, anyone, let me confirm
Uh,
I am clearly
please.
audible.
Yeah, you are audible.
Okay, sir, thank you. Assalamu alaikum, everyone, and a very warm welcome on behalf of Vitanova International Alliance for Sciences. It is my pleasure to welcome you all to the fourth session of Bioinformatics and Molecular Docking Online Internship Program 2026. I hope you all are doing well and enjoying your learning journey so far. We are pleased to see your continued enthusiasm and participation throughout this internship. Today's session is another important step toward developing practical expertise in bioinformatics and computational biology. Before we begin, I would like to make an important announcement regarding attendance. A Google form will be shared in the live chat during the session. All participants are requested to fill out the form carefully to mark their attendance. Now it's my great pleasure to introduce our esteemed speaker for today's session, Ms. Abeera Iftikhar. Ms. Abeera is a silver medalist biotechnology graduate from Rifa International University and the founder of BioCode Innovators. Her expertise lies in biotechnology, bioinformatics, and computational biology. With experience in research projects involving molecular docking, protein structure analysis, and computational drug discovery. She is passionate about mentoring students, promoting scientific learning, and helping aspiring researchers develop practical skills in biotechnology and bioinformatics. Today, she is going to deliver a session on protein structure validation, a crucial aspect of structural bioinformatics that helps assess the quality, accuracy, and reliability of protein models before they are used for downstream applications such as molecular docking, drug discovery, and protein engineering. We are honored to have her with us today and are confident that her expertise and insights will provide valuable learning opportunities for all participants. Without further delay, I would like to invite Ms. Abeera Iftikhar to begin today's session. Ma'am, the virtual floor is all yours. Please take start. Thank you so much.
Miss Abeera Iftikhar, am I audible to you?
Okay, participants, I have a conversation with Ms. Abeera. She is facing some issues. She will join shortly.
Everyone, stay in the room. So, our instructor is joining within 1 to 2 minutes. Soon we will share the link, attendance link in the chat, live chat, and you have to mark your attendance through Google form. Okay, and one last message. After that short presentation, there will be a practical lecture regarding the protein structure validation. We will learn some different new tools about protein structure validation. So stay in meeting. Hope for the best.
Assalamualaikum everyone. Am I audible enough?
Walaikum salam. Yes, miss, you are clearly audible. Please continue.
Thank you so much. Assalamualaikum everyone. I am Abeera and I am the founder of BioCode Innovators. Before we begin, I would like to thank Vitanova International Alliance for inviting me to be the part of this session as an instructor, and I'll be the instructor for today's session, and I'll look forward to sharing up the knowledge and having an engaging discussion with all of you. Let's get started. I think so. You have I suppose you all of you have learned so much through the program. Today we will be covering up this protein structure validation. I hope so you had covered up how to download the protein 3D structure. We will be going for the structure validation, as the structure validation is really important for the further analysis. Whether we are predicting the structure from any of the prediction tool like iTaser, AlphaFold, Swiss model, or any of the Galaxy Refine model, or either we are going to validate the already existed structure for the further analysis. As for the most important one and further analysis is docking. If we are going for the docking with any kind of particular protein, we need to validate the structure before moving forward. And if the 3D structure of that particular protein we are going to work doesn't exist, then we have to move toward the prediction of that 3D structure. Afterwards, we have to move toward the structure validation. So it is the most important step for the molecular docking. So either you want to validate your structure, if your structure is reliable enough, or either you want to check if your structure is good for the further any kind of analysis. It will be dealt with the protein structure validation wing. There are multiple tools. We are going to operate with the project, ERRAT, and RC plotting. The most important one results for the structure validation are considered for the ERRAT and RC plot. As let's say, whenever we are going to work for any kind of further analysis, let's say we are going to work for the docking and we are operating any kind of protein with the ligand. So there is one question that came up in your mind, is that is my protein model is acceptable, or is my protein model is structurally validate enough to be used for any kind of further analysis? So we are going to cover up this one. Firstly, we are going for the downloading of 3D structure of protein from the PDB site. PDB is a database. We are going to cover that up. I think the candidates have covered this up. Right, Ms. Hafsa? So I don't have to move toward that way. I will directly move toward the validation.
Yes, miss. Please continue.
Okay. There is a question why we have to go for the validation, like why validation is needed. any kind of predicted structure or any kind of structure, the 3D structure of protein we are going to use for the further analysis, we have to check if there is any kind of incorrect angles, bond angles, any kind of steric clashes, or any kind of incorrect folding, any unusual residues environment, and any kind of unstable conformation. So we have to fix that, and we can check that wing, all of these things using all the refinement and using the validation wing. Validation tools detect all of these problems. So um, we can say that protein structure refinement and validation are closely related. If the structure validated results aren't good, we have to refine that structure too. Then again, we have to move back toward the validation result to compare up the pre-refinement results and the post-refinement results. So we can select up the reliable 3D structure of protein for the molecular docking and the drug design. Next slides, please. So that's why the protein structure quality matters. We have to evaluate it, whether the protein model follows up the particular geometry rules, or either the structure is experimentally good enough with the stable conformation and steric clashes. And if there is any kind of clashes with the model, we have to fix it using the refinement way. But right now we are going to cover up the validation, like if the protein of ours with the 3D structure is already here over to the interface of PDB, then we have to refine, we have to validate it using RC plotting and ERRAT. These two things are considered as the good one according to the scientific work, and we can easily and write it over to our research paper. These are considered as the validated results for the research paper. Next slide, please. Next slide, Ms. Hafsa. Thank you. So ProCheck is also considered as the tool for the validation. And this wing we will go for checking up the stereochemical qualities of our protein structure. And we can say that it is a widely used tool for the validation, and it evaluates our stereochemical quality of our particular protein by analyzing its geometry, any kind of comparing it with the high quality experimentally determined, already determined structures. So let's say, what does ProCheck analyze? If there is any kind of question like that, like why we are going for the ProCheck, what does ProCheck analyze? So ProCheck analyze the two most important things. The major one wing is considered as the ProCheck, and the RC plot and the ERRAT is the sub wing of this ProCheck tool. Okay, so we are going for the RC plotting afterwards and the ERRAT afterwards. We will go for checking up the bond angles and the dihedral angles along with outputs from the Ramachandran plot and other residue studies. Next slide, please.
Here is next slide, RC plot introduction, please.
Okay. Um, RC plotting is considered as the minor tool, but the validation results for the RC plot is considered the good one result for the scientific purpose, as we can easily cite it in the research paper of ours. For that, we are going to use it for the docking and any kind of other workflow lines. So there are two ways or the two angles that represent the RC plotting. And there are many regions there are four type of regions for the RC plotting. And there are two angles we can check as a backbone. One is a pi angle and the psi angle, and we have to immediately check and rely on the allowed and the disallowed confirmations. Like if the protein major regions are falling in the allowed regions or in the disallowed region. There are four particular as my mic muted. So Ramachandran plot will identify all of the allowed conformations or the favored regions. If the protein 90% the protein regions are falling into the favored region, then the protein result will be above 90%. Then and the above 90% results are considered good one for the further analysis. And we are also going to check for if the major of our protein is falling in the disallowed region. If it is falling in the disallowed region and the results aren't good, then we have to move toward the refinement of that particular structure of protein. Yes, next slide, please. And with the RC plotting, we will check for the stereochemical qualities and the bond angles with the dihedral angles, and the output results will be the residue plot. That is the most important one, and the residue statics that we can read it out, but it isn't something that we can write in the research paper of ours. Next slide, please. Besides this is the most important thing. Your RC results, you have to keep that in mind. There are four major regions. One is considered as, or we can say the four portions of one square or one rectangle type graph. And these four regions are highly considerable for the result prediction, as the most favored regions have ideal backbone conformation. If our protein falls in the most favored region, then we can consider it as a good protein. It will give us a good RC plot result. And additionally allowed region, it is also an acceptable region. And then there is a generally allowed region. This is also acceptable, but may indicate any kind of flexity. Just it is just less common. And the disallowed region is the problematic residue, or we can say that the protein, if the protein is falling majorly in the disallowed region, then it is considered as structurally unstable and incorrect one. There will be any kind of modeling errors, maybe poor refinement, and maybe incorrect folding. So next slide, please. So let's say, how can we predict or interpret the results? Residues of our protein must be falling into the favored region above 90%. Like high quality model, it says that the 90% residues are falling into the favored region. And the poor qualities means model means the many residues are falling in the disallowed region. And we can't use the model that is majorly falling into the disallowed region for any further analysis. And if there is any kind of going like that for the poor quality model, so we have to go for the refinement. We have to move the residues from the disallowed region, and we have to change up the confirmation to the good one, so we can get the good results. So generously allowed regions will hold up only the model quality of 1%, and the disallowed hold the major or the poor quality. Besides this, we can also say this criteria. If the favored regions are considered as the excellent one version of the protein, in a favored region above 90%, and if it is 85 to 90% in the favored region, then it is considered as good one. And if it is less than 85%, then it is considered as the poor one result if the protein is 85% falling in the favored region. And if the protein is in the disallowed region, if the protein is 0% falling into the disallowed region, if our protein is 0% falling into the disallowed region, then the protein is good one. And if it is majorly falling into the or beyond 2% or 10%, so it is considered as the poor quality model. For example, I am giving you a case study. Okay, just give me an answer in the chat box. Like, let's say my protein have a structure or the model of my protein is falling into the favored region at 95% rate, and in the allowed region at the 4% rate, and in the disallowed region at the 1% rate. What should be the results? Like, how to interpret the results? Let's say, will the result or the structure is excellent one, or is it the good one, or is it the poor one? Do I have to type it? You can go for this case study. I will give you clear map before move toward the practical one session. Anybody who can answer it? If my protein is falling into the favored region by 95% and into the allowed region by 4 to 5% and in the disallowed region by 1% or 2%, as my protein structure is good enough, or the excellent one, or the poor one? Okay, we will take the answers at the last. Then ProCheck. ProCheck is basically the major wing through which we can generate the RC plot and the bond length and the bond angles and any kind of qualities. Next slide, please. Then there is an ERRAT tool. ERRAT is considered as the most important one for the further analysis of the validation wing, as it evaluates any kind of non-bonded atomic interactions, like if the amino acids aren't aligning a good way, or if there is any error exceeding the limit of our warning area. So ERRAT goes for that wing. Besides this, we also detect any kind of error. If there is any kind of error in the protein backbone, like if there is any kind of error in the amino acid interaction or atomic interaction with each other, or with the folding way, or any kind of side chain environment interaction. So overall quality factors depends if the results are good enough or not. Besides this, what does ERRAT check? ERRAT checks for the atomic interaction, like in the carbon to carbon and carbon to nitrogen, from nitrogen to nitrogen, and carbon to oxygen, these type of interactions, to check for the high resolution experimental protein. Next slide, please. And this is considered as the important one wing. You have to keep in mind whenever you are working with the ERRAT, the results matter more than anything else, as if the results are above 90%, ERRAT results or ERRAT score is above 90%, then the structure is excellent. You don't have to go for refinement. You can directly go for the further analysis anyway, if you are going for the docking or anything else. And if your structure ERRAT results falls in the range of 80 to 90%, then the results quality is good. And if it falls less than 80, then it requires some of the refinement. but the highest ERRAT score is considered the better structural reliability or structure quality for the further analysis. And if your results are falling very low, like close to 50 or below to 50, then the protein is unstable or unreliable for any of the further analysis. It highly needs refinement. Besides this, one more question for everyone. As let's say my protein's ERRAT score is 95%. Is my protein good enough? Ms. Hafsa, you can move the slide. Is my model acceptable? And if my protein score for in like 60 to 70, is my protein good, is in a good quality or the poor quality? Does it need any kind of refinement like that? Next slide, please. So these are the workflow. We are going to go for this validation results. Checking like this. I haven't downloaded any protein. We will download it and we'll go for the validation results. We have to obtain the protein CD structure, and we will validate it using the project. Project will have the sub wing as RC plotting and the ERRAT. And we will be going and analyzing the validation scores and identifying if there is any kind of structural errors or any kind of outliers. And either we have to improve any kind of stereochemical qualities or anything like that. And we have to select for the practice purpose, you can select up the protein that shows the good ERRAT results, or basically the protein that shows the high resolution shows up the good result for the ERRAT or the project for any kind of downstreaming. So we can say that if any kind of refined protein we are using or any of the protein 3D structure, we have to validate it using the project and the ERRAT or the RC plotted analysis. If the RC plot reveals the result within 90 to above range, then and in the most favored region, and additionally all of these indicating acceptable stereochemical activities or qualities, or ERRAT shows up the results above 90%, then we can go for any of the further downstream analysis with that particular protein. Next slide, please, Ms. Hobson. Yeah, that's all for the overview. And as we are going to take up the major step and the most important one, you can skip it whenever you are moving toward the molecular docking or any kind of further analysis, as it will give you, or it will give the others a proper guide as if your structure was validated, or where was the validation result of yours before moving toward the downstream analysis. So make sure whenever you are going to write something like report or the paper, make sure you don't skip this step, as the protein structure validation is really important for the downstream analysis. Like if you are writing up any kind of paper and your professors ask you where is your validation result, and you have skipped this point, that saying like the protein already have the good results, so it doesn't work this way. Okay, thank you. And Ms. Sapsa, let me know how to share up the screen. We'll go for the practical one.
Miss, there is an option in the screen that shows the present. You can click and it will show the options.
Okay, thank you. Just a minute.
Then you can go for the screen sharing.
Okay, just a minute. I'm sharing it. my screen is visible, right?
Yet not.
Not yet. Just a minute. It's I think it's visible from my side. It's visible from my side. I think you have to update it.
Let me check, miss. 1 second. Yes, now it's visible. Yeah.
Okay. Okay, let's move toward the practical session, and then afterwards, if there is any just like as for as Ms. Hafsa said, you had covered all of you had covered the PDB database. So all of you knows everything about this PDB database, and we are just going to download up the 3D structure of protein, and we will be moving toward the structure validation. Before this, keep that in mind. If the resolution is in the range of 1 to 2, oh sorry, 0 to 2, it's good. You don't have to go for the refinement before moving toward the validation. If your structure falls in the area of 2 to 4, you do have to move toward the refinement before the validation. Even if you go for the refinement first, for the validation first, your results won't be good. You still have to move toward the validation link. Let's go for the protein structure validation. You can directly search it by using the protein structure validation, or you can even search it simply writing up the project RC-plot or ERRAT. This server offers all of these tools in one go. I have to here it offers many tools. ERRAT, Verify 3D, it isn't considered the good one. These two, ERRAT and ProCheck, along with the RC results, are considered the good one for the scientific writing. So just a minute, we will download it. Here is how you will download the 3D protein structure. I don't have any kind of visualization software in this laptop, but it will work this way. Okay. You have to choose the file for this interface. Okay. Just a minute, everyone. Ms. Afsar, yeah, can you approach me on the website?
Yes, miss. Sure.
Just, one more thing. Just freeze up my screen. I have to open up my WhatsApp.
You can stop sharing or just share one folder.
And stop sharing my screen and share it in a minute. Um, Actually, I don't have any kind of visualization software in this laptop. The other one isn't working properly right now.
Miss Abeera, please share the screen. I will take it on board.
Okay, I'm sharing it back. Um, this one laptop doesn't hold any kind of visualization software, and the other one isn't working right away. So I am not able to go for the PDB structure. The rest is good. Right away, it needs the PDB structure, and the interface offers the CIF way. Can I log in from my other laptop too? If it
Yeah, sure,
works this way?
sure.
Just a minute.
Everyone kindly stay in the room. So we are sharing the screen from an other device. Thank you so much, everyone, for your cooperation. Uh, Miss Novo Akhtar, are you there? Am I audible to you?
Yes, you were there.
Okay, please share me the attendance Google form in private chat in in the live stream.
Okay. Yes, miss, you can join directly from another laptop.
Okay, just a minute.
Just use the same link I had shared for joining this one.
I'm signing off from the previous screen and I'm logging to this new screen. Is it visible now?
Not yet. Okay, now it's visible.
Okay, just a minute. We'll have to go for the protein structure validation. And the protein is in the CIF model, and it's just a minute. Let me open it up in the Discovery Studio. Here is the Discovery Studio. You can also open up your files in Discovery Studio as PayMol and RASMols are also considered as the PayMol and the RASMol are also considered as the software for the validation, for the visualization. But here we can also check for any of the protein structure using the Discovery Studio. Okay, so besides this, we can also see that right away this protein holds many of the small dotted lines or dotted points that are considered as impurities for the protein. Whenever you are going for any kind of docking analysis, you have to remove all of these. You have to clean up your protein too. So in this way, we also can save this as a PDB file. Otherwise, if you don't save it like that, it still will be opened up in a Discovery Studio file. Here, let's move toward the practical one version. And we have to upload our file. Choose the file, and you can upload your Discovery Studio file too, and you can also upload your RASMol or the PayMol visualization PDB file too. You have to upload it. You can select up the name. I'm so sorry, it still isn't working. I don't know what is the problem right away. PDB file. Let's save it again. Yes. Let's do it one by one and see which one is the PDB file now. And do download your RASMol or the PayMol for the visualization. It is now the PDB file. You simply have to go for the Discovery Studio. If you don't have any PayMol or the RASMol software, Discovery Studio is also a software that is used for the docking purpose. And you can also save up the file of yours or the protein of yours in a PDB version from here. Click on the file. From this drop down menu, save up your file, and here from this drop down menu you have to check for the protein data bank file. And here, let's go back, and this will go for the run program. Here it offers us the new interface, and this will go for the 4 to 5 interfaces. We are going to check for the ERRAT results, and we are also going to check for the project results. So let's start with the ERRAT result. As we have already uploaded the protein, we don't have to upload it separately for all of the working folders, and we can go for the same protein and one upload to check for the ERRAT results and also the project results. ERRAT result is 100%. That's good. If the ERRAT result is above 90%, then the protein is considered as a good one protein. And it also shows us this graph. This graph isn't in a full way, otherwise the graph was let's see, the graph isn't the expanded one version. Okay. These are the log values, but we don't have anything to do or do with the log values. By the way, the results are 100%. The results are good enough, but the ERRAT graph isn't visible completely. As for the graph should be the longer one, and it shows us the warning ways. We can also go for the other way around. Let's go for the ERRAT directly. Might be possible it will offer us another interface. Here, like this. This one graph is considered the validated one for the work purpose. It brings us back to this interface again. But I think the protein is having any kind of problem. The sample protein we are using is having any kind of problem. Still, the results are good with the 100% protein. And make sure whenever you go for any kind of further analysis, keep in mind that the results should be falling majorly into the non-warning region, and that is the reliable results. Here again shows us this graph isn't showing us the lengthier one graph. Besides this, there is a project result, and the project offers us the interface with the RC plot, and RC is considered as the major one for any of the further analysis to check up the validation if the protein is validated enough or falling into the region of most favored region or not. It will give us the results back in few seconds. Seconds and the result will be shown to us. Till then, if anybody have any question, drop your question into your chat box, and we will answer them at the last. Come on, why is taking so much time?
Miss, there are some questions in the chat. Should I read for you or you can go for them?
At the end,
Um,
we will discuss the question. At the end, please.
okay, we can take the questions at the end. Maybe I don't know why it is taking so much time. Might be the protein as the heavier one.
If it is taking some time, so we can discuss some questions as
Okay,
well.
go ahead. I'll also search for another protein that will give us the quick results. Yeah, until then, we can also take the questions.
Okay, should I read for you?
Yes.
Okay, someone is asking which validation parameter is most important before proceeding to molecular docking.
Validation parameters, both validation parameters, along with the RC plot results of yours and the graph that it will offer as a result. You have to plot it or place it over to your um, report or whatever scientific writing you are doing. And besides this, also the ERRAT graph is considered as the good one for your working into your report or the scientific research paper of yours.
Okay, here is next question. What is the difference between ERRAT, word check, and pro check?
I didn't get it. Sorry.
What is the main difference between ERRAT, word check, and pro check?
Project and
ERRAT.
ERRAT and Project both are considered as the validation tool. There isn't any kind of major difference, as the difference only lies in what this both of these tools are validating separately. As for the ERRAT, it will offer us the way like our protein is it falling into the majorly allowed region or not, or the ERRAT one shows us if the protein atomic structure or interaction is good for or staying into the less warning region or the more warning region. That's
Okay, thank you, Miss. Here is next question. Can we use UniProt database for protein structure validation?
No, for protein structure, I just clear up this question. There is one more thing. The protein or the UniProt is a database through which you can get any kind of annotated version of your protein. Annotated version is considered as the version that give us all of the information about that particular structure. But whenever we are going for docking or the molecular analysis, move toward the PDB interface because PDB offer more reliable 3D structure of protein. And for the refinement is another wing, and and we have to operate it from the another interface. It Uniprot doesn't offer any kind of refinement. And if you are asking about like using the Uniprot 3D PDB structure or 3D structure, it isn't reliable enough as compared to the PDB one.
Okay, thank you. Here is next question. How to read RC plot?
Your RC plot, we will look into this RC plot just in a minute. As the results aren't visible, still it's taking too much time. Um, I am signing off from my screen. I think I have these type of results in my laptop. Just I will come back here.
Yes, you can show these results so students can get the idea how to read this.
Here I see.
Students, still that the Google form is shared in the chat. Many times you can mark your attendance.
As my screen visible back.
Yes, it's taking loading. Now, yes, it's visible.
Okay. This type of graph it offers if your protein is the heavier one. Sometimes it takes more than 5 minutes, but if your protein is the hold lower disrupted residues, it covers up in few seconds. Our protein is taking so much time, so we are shifting toward the understanding of this plot, RC plot. Um, this is the plotting result, or this is the major wing that you can place directly into your research paper or your research report. And these are considered as the four major wings that I have already explained. That one is the major one, allowed region. The first one is considered as the most favored region. The and next onward is considered as the allowed region, and this one is the generously allowed region. And the this one last one is considered as the disallowed region. Make sure whichever whatever protein you are using, it falls into this disallowed region as less as possible. So this graph also offers these results. Like right here, if I see this is one of my working file, and the residues wasn't good enough, and like in the most favored region the protein is 69%. So if I am going to work over the molecular docking with this protein, uh, I have to move toward the refinement before moving toward the for the docking purpose. So here it offers us all of these results in a values. Like here is the favored region results. My protein is 69% into the favored region, and into the additional allowed region, that is this one wing, and it is just 18%. um, this is also much higher than the ordinary one. And from the generously allowed region, my protein should be just 2 or 1% into this region, but here my protein shows it is 7.7% in this region. So basically, in short, we can say that I have to refine this protein before moving toward the docking purpose. But if my results are falling into the favored region above 80% or 90%, then I don't have to move toward the refinement. So it also offers us the random these residues, the known glycine and the known proline. These aren't considered for any kind of further analysis, but these all of these plot results and the statics are considered highly considered for any kind of further results. Besides this, I think I also have it at value results. This was the protein result, but just a minute, let me see. This one protein wasn't good. This one is also the ERRAT result. Whenever you download any kind of ERRAT result in your system, it offers this type of screen. As for if you open up into the interface, it offers you the wide graph, but whenever you download it in your system, it offers you this type of graph. And make sure the protein relays lower than this yellow line, as this yellow line is considered as the warning line. And if your protein is majorly crossing up this warning line, then you have to move toward the refinement purpose. These are also the ERRAT results, and here it shows, I don't know if my screen is visible. Whenever you download any kind of these graphs, it offers this way in your laptop, like in a blank way, with all of these red lines that shows us my protein is highly unconsiderable or unstable for any kind of further analysis. As for the results, quality factor results are 38% or 38 point something. That isn't considered the good one. If my factor results crossing up the 8% results, sorry, 80% or 90%, then it is considered as a good one. Like if my protein relays in the ERRAT result with this yellow color, or it stays below this 99% line, then it should be, or we can say that it will give us the results that will cross maybe 80%. But right here, all of these red lines and the full graph resemble that our protein quality isn't good, and we can go with 38 point something, like quality factor for the further analysis. We have to move toward the refinement. I think that's all. These are all of the working. Here is considered as, we can also say that the protein quality factor is 93%. That is considered as the good one. That's all from my side. And just let me
Okay,
know, Miss
thank you.
Upside,
Yes,
anything left
miss.
over.
Yes, it's all clear from my side. So should we take further questions?
Yeah.
Okay. Okay, guys, if you have any questions, still you can write in the chat as Miss Abeera is here, so we can address your questions. Till that, I see two or three questions I can ask Miss Abeera that. Here is a question: how do you justify a crucial catalytic residue falling into a disallowed region of a RC plot?
So sorry, Miss Hafsa, your voice is dropping. Can you write it in the chat box?
Ye-
Your voice is dropping off.
Here, okay. Can you see the question on the screen now? It is
No,
on
there
board.
isn't. No, there is no screen. Or I'm sorry. Wait a minute. Should I move toward the comment section?
Uh, yes, comments are question is visible on the screen now. The screen where presentation is showing.
Okay. From these tools, which one is the best suited or particularly in the agriculture sciences? Even if you are moving toward the agriculture sciences or you are working with the human genetics or the health wing, both are considered the good one. If your protein, whatever you are using, or whatever thing you are covering. As for the agriculture, if you are going for the agriculture bioinformatics, you still would have all of these sequences or 3D structure of your particular protein. Either it belongs to any kind of stem tissues or any kind of particular plant, then it is considered the good one for anything related to bioinformatics if you are moving toward any kind of further analysis. So, how do you justify this crucial catalytic residue? For the disallowed region, if your protein or the structure results are falling more than 1% or more than, we can say that the range is 1 to 5%. But if all of these ranges are clearing up, like keeping up in a way, the protein is falling in 1 to 5% or 1 to 2% in the disallowed region, then you can say that your protein isn't good enough. But if but also one more thing, if the protein of yours also showing up 90% or 95% result in the allowed region, you can take it for the further analysis. Anything else, Ms. Hafsa?
Yes, next question is on stage. When
um,
RC is,
When
can you
RC
see
is
it?
also good, but the React score is poor, which should be prioritized. RC is more prioritized as compared to the ERRAT, but if the ERRAT is, let's say your RC results are falling into the region of 90 to 95% or 85 to 90%, and the ERRAT results is also falling into the 80% range, then you can take both. But the more considered one or the reliable one is a RC plot because it offers us the most reliable or the detailed graph that we can place it in the paper and cite it directly. How would you design and validate composite structure quality index using the weighted contribution from the? What kind of question is this? I didn't get it. Your question is okay. Where is the question? Can you show the question again, Miss Hafsa?
Yes, here is the next question. How to differentiate between different protein structures like beta sheets, right and left alpha helix in RC plot?
You can identify your alpha helices or beta sheets from the Ramachandran plot using the secondary structures like does it occupy any kind of phi or the psi angle regions. Like if it is falling in the approximately into the phi angle regions, right-handed alpha helix, or the approximately into your 45-degree in your psi angle, you can consider it as the lower left quadrant for that plot. And same way, if for the beta sheet you are going for, so the phi angle and the psi angle qualities are different, but you can check up the upper left quadrant. Anything else, Ms. Hafsa?
Students, if you have still any questions, you can write in the chat. I think all are addressed till now.
Okay, thank you so much. Can I sign off from the meeting now?
We can wait for 1 or 2 minutes. If students have any
Okay.
question, then we address and then we can end. Okay, students, please be quick if you have any questions. Okay, here is a question. Can you see, Miss Abeera?
Yes.
Okay.
So we can say that RC is used to check the quality and ERRAT is used to check the bonding in the structure. Yes, RC is used to check for the quality if the protein is falling in the most favored region or not, or either the protein is good for the further analysis with the good ERRAT, good RC results, or ERRAT will give us the bonding result if the amino acids bonding are good with each other or the atomic interaction is good for, or is it falling into the favored region or is it falling directly into the unfavored region. In drug design, results of RC or ERRAT both are preferred. Whenever you are going for the drug designing, the results of ERRAT and RC results of yours will be posted or pasted in your work or the paper you are writing or the report you are writing. Both are considered the good one. Can we use verify 3D? Verify 3D, you can use it for your purpose, but it isn't validated enough or considered the good one for the any kind of research paper analysis. And work check, I haven't used it personally because it doesn't offer any kind of good result, and results of work check aren't reliable enough. Yes. Hafsa, you can share up the name of all of these questions. Oh, sorry, the tools in the chat box. Okay.
Yes, ma'am, I will share.
Okay. Anything else? Um, I think that's all from my side. Um, without MD simulation, yes, without MD simulation, your result or your docking project isn't completed or is considered as incomplete. But that is not my wing. The other instructor will be covering up this MD simulation topic. All done. Okay, one more question. I didn't get the last one question.
Yes, someone is asking what is the mechanism behind RC plot and ERRAT plot.
We can say that RC plot validate if the protein structure or by analyzing its fibon or the torsion angles or either identify whether the protein amino acid residues are lie in the statically allowed region or the favored region or in the most disallowed region. Besides this, the ERRAT it offers us the results by analyzing the non-bonded interactions and comparing them with the experimentally determined any type of proteins or the proteins that already have the determined type of good ERRAT result structures, and the highest result scores we can say that there will be a fewer structural errors. Why are some residues allowed in the disallowed region? We aren't the one that are allowing the residues in the disallowed region. It is protein that is itself aligning it into the favored or unfavored regions. That's all, Miss Afsa.
Yes, I think it's all clear. I think no further question in the chat.
Okay, thank you so much, everyone, and
Uh, th-
thank you so
Thanks.
much
Thank you. Thank you
for
so much, Miss
allowing me
Abeera.
in the discussion. Thank you.
Yes, we are also thankful to you for
Okay,
such an insightful session. Thank you so much. And guys, I
let's
hope
meet
you all have
in the
marked
next.
your attendance via Google Form. And so that's
Uh,
towards the end of the session.
Miss Assa, please conclude it. Also, tell a little bit about the report. Report will be submit at the end. We also already shared report sample, and we will also share a new sample report in the group. You have to make your report according to sample. Thank you so much, everyone, for joining us.
Yes, sir. Special thanks to our guest speaker, Ms. Abeera, for such an insightful session. And we are also thankful to all the participants for joining us today. And I hope you all you all had learned something new regarding protein structure validation, and very enthusiastic about our upcoming lectures. So that's all for today. About report, the sample is already shared in the group, and further we will share another for your reference. And that's all for today. And I hope you all had marked your attendance via Google Form. And one last thing about the attendance, that if you are having any queries regarding attendance today, we will address them after tomorrow's class. So I will share a little detail about it in the group, and I hope you will get the idea. So let's end today's session. Thank you so much, everyone. Okay, sir, you can end the session now.
Okay. Goodbye, everyone. Thank you so much, Miss Abira and all other participants. Have a good day. Bye bye. Take care.
Thank you, sir. Allah Hafiz.